Hello students, today I am going to take the meiosis topic, and I am Geeta, botany faculty. So in the cell cycle we have completed already mitosis, and then we are going to deal with the meiosis. Meiosis generally it occurs in the reproductive cells or germ cells, mainly reproductive cells it occurs. Generally in the formation of gametes, gamete formation it occurs, or spore formation. It always occurs in the diploid cells only. There is no meiosis which will be happening in the haploid cells. It always occurs in the diploid cells only. So diploid cells ultimately by the end of the meiosis they will be converted into haploid cells. After the meiosis, one parent cell, one diploid parent cell will be forming 4 daughter haploid cells. So this occurs in diploid cells and it forms 4 haploid cells after the meiosis. So meiosis always it occurs in the diploid cells only, and always it results in the formation of haploid daughter cells. Now let us see the meiosis which occurs in the reproductive cells. It is of 2 types, Meiosis I and Meiosis II. In the Meiosis I, you can see here, Meiosis I is again divided into Karyokinesis I and Cytokinesis I. So in meiosis, basically one cell will undergo 2 times nuclear divisions and 2 times cytoplasmic division. So Karyokinesis I and Cytokinesis I, nuclear division and cytoplasmic division once, and later Karyokinesis II and Cytokinesis II will happen in the Meiosis II. So it is basically divided into Meiosis I and Meiosis II. Meiosis I is further divided into Karyokinesis I, Cytokinesis I. Meiosis II is subdivided into Karyokinesis II and Cytokinesis II. In the Karyokinesis I, again this is subdivided into prophase I, metaphase I, anaphase I and telophase I. By the end of this, the Cytokinesis I will happen and it will result in the formation of 2 daughter cells. Meiosis II, in Karyokinesis II, again you have the subphases, prophase II, metaphase II, anaphase II, telophase II, and finally you will be getting the formation of 4 daughter cells. These 4 daughter cells are always haploid, and the initial, whatever the cells that are there, they will be the diploid cells. By the end of the telophase itself, these each daughter nucleus will become the haploid daughter nucleus. So because of this, meiosis I is called as reductional division and meiosis II is called as equational division, because 2n is becoming n in the meiosis I only. So this is reductional division. And in meiosis II, n cells, the cycle is starting with the n cells and it results in the formation of daughter cells which are haploid only. So this is the equational division. So meiosis include 2 steps, meiosis I and meiosis II. Meiosis I is the reductional division and meiosis II is the equational division similar to mitosis. Let us see the next one. Meiosis I. In this, Karyokinesis I and Cytokinesis I is there. Karyokinesis I is subdivided into 4 phases, prophase I, metaphase I, anaphase I and telophase I. Prophase I, majority of the events will happen in the prophase I only, because prophase I is the longest phase of total meiosis. The longest phase is the prophase I because it is subdivided into phases. We will see the prophase I subphases after this. So by the end of the prophase I, you know that chromosomes will be formed, nucleolus, nuclear envelope, Golgi, ER disappears, centrioles would have migrated towards opposite poles, and the chromosomes must have scattered in the cytoplasm like the mitosis. But this is again further subdivided into 5 phases: leptotene, zygotene, pachytene, diplotene and diakinesis. In this metaphase I, whatever the chromosomes that got released here into the cytoplasm, they got attached to the kinetochores and the spindle fibers and they are placed on the metaphase plate. That homologous chromosomes are placed on the metaphase plate, and then anaphase I, the homologous chromosomes of the bivalents get separated towards opposite poles, and in telophase I, you can see here that the 6 chromosomes are here in this anaphase I, but 3 chromosomes would have migrated towards opposite pole. So here you are getting 3 chromosomes in each daughter nucleus. And after that, cytokinesis will proceed, and by the cell plate formation or cleavage furrow formation. Here it is a cleavage furrow, so it deepens towards the center and it divides one parent cell into two daughter cells. And these two daughter cells are actually haploid. You can see down below the chromosomes condense and the nuclear envelope breaks down, and a major event which happens in prophase 1 is crossing over occurs. Here, centromere, the kinetochores are there, the spindle fibers are coming from opposite poles, and to this the spindle fibers are getting attached. You can see that the homologous chromosomes are attached towards the poles of their side only. So each chromosome is connected to only one pole, not to the both the poles. One chromosome is always connected to the one pole only in the meiosis I, because two sister chromatids are connected to two spindle fibers coming from only one pole. That is the difference. And homologous chromosomes move towards opposite poles. That happens in the anaphase I. Here they are arranged in the form of bivalents and they get separated here and they move towards the opposite poles. In this first cell, we have started with 6 chromosomes, and ultimately in each daughter nucleus we are getting 3 chromosomes. So this division, it is called as reductional division. Because basically here 6 chromosomes are there, and here in each cell we are getting the 3 chromosomes. So it is the reductional division. Now let us see the prophase 1. Prophase 1 is the longest phase of the meiosis totally, and it has 5 subphases starting with leptotene, zygotene, pachytene, diplotene and diakinesis. In leptotene, basically the chromosomal condensation will begin, and chromosomes will be appearing like bead-like units on them, and these beads are called as chromomeres. Beaded-like structures will be there in this, and they are called as chromomeres. Condensation begins in the leptotene. Then in zygotene, the 2 homologous chromosomes which are coming from mother, maternal chromosome and paternal chromosome, they come towards each other because of an attractive force between them that happens because of synaptinimal complex proteins. And the 2 chromosomes which are identical, they will come together with the help of synaptinimal complex proteins and they pair up together. The process of pairing is called as synapsis. And the end result of these synapses is the formation of the bivalents. Bivalents are formed. Bivalent is nothing but 2 homologous chromosomes. And the process of pairing is called as synapsis. That will happen in the zygotene. In the pachytene stage, here though the chromatids are 2 from the S phase onwards, due to the condensation, they can be very clearly seen in the pachytene because the condensation begins in leptotene and it is continued throughout the prophase 1. So in the pachytene stage, the condensation would have progressed a little further, and we can see that the chromatids get separate, untangled here, and we can see that each chromosome will be having 2 chromatids each. So here bivalent means 2 homologous chromosomes. Now 2 homologous chromosomes of a bivalent will be showing 4 chromatids each in pachytene. So that 4 chromatids containing bivalent will form 1 tetrad. This is also equal to 2 chromosomes, 2 homologous chromosomes. So they are seen in the form of tetrads in the pachytene stage. And this is the longest phase in prophase 1 because it will take more time here. And in this pachytene, the actual process of crossing over occurs. The crossing over is nothing but the exchange of genetic material between the 2 non-sister chromatids of a bivalent. Bivalent means 2 chromosomes will be there, homologous chromosomes like this one and this one, this one and this one. Now they exchange their genetic material between the non-sister chromatids. That means these 2 or these 2. They will be exchanging their genetic material. That happens by the formation of recombination nodules. Here you can see that dark color line is there. Recombination nodules are there. And they exchange their genetic material with the help of an enzyme called recombinase enzyme. So by that process of crossing over, they would have exchanged their genetic material, but still they remain contact in the packaging. So this is the important step in the total prophase 1 because it leads to the variations in a species because they would have exchanged their genetic material due to crossing over. Next, diploty. This stage lasts for months or years in some oocytes of vertebrates. Not in all, but it lasts for months or years in some oocytes of vertebrates. Other than that, here the synaptonemal complex which was here, which is holding the bivalent chromosomes together, that will be dissolved here. So synaptonemal complex get dissolved here. So the attractive force between the two chromosomes will be dissolved and they tend to separate. They try to move away from each other in diploty. And the process of condensation already started here and it will proceed in the same way. That will happen in the diploty. When the chromosomes are trying to separate away from each other in the diploty, the X-shaped structures are seen clearly here. They are called as chiasmata. You can see here chiasma singular, chiasmata plural. This chiasmata here, they would have reached towards the ends of the chromosomes. This is called terminalization. Terminalization of chiasmata occurs in the diakinesis. That means chiasmata are actually seen clearly in the diploty. They are formed in the packaging, but they move towards the ends of the chromosomes in the diakinesis. Terminalization of chiasmata will happen. And in the same diakinesis, this nuclear envelope, nucleolus disappear, Golgi disappears, ER disappears. The centrioles would have migrated towards the opposite poles. All the events of prophase will be finished by the end of the diakinesis. As the nuclear envelope disrupts, here all the chromosomes will be released or scattered into the cytoplasm. So basically in leptotin, chromomeres are seen and process of condensation begins. Zygote, process of pairing starts, synapsis starts because of synaptonemal complex proteins that results in the formation of bivalents. They pair up each other. In pachytene, tetrad stage it is called because each bivalent will be showing the tetrad, 4 chromatids. So in this tetrad stage only the crossing over occurs, the genetic exchange of chromosomal material between the two chromosomes. That happens here and it leads to recombinations and sources of variations that happens in the pachytene. In diplotin, the synaptonemal complex will be dissolved and they try to separate away from each other, and due to that we can see the points of crossing over X-shaped structures called chiasmata in the diplotin. In diakinesis, terminalization of chiasmata happens and all the events of prophase 1 will be completed, like nucleolus, nuclear envelope, Golgi, ER disappears. Centrioles would have migrated towards the opposite poles, spindle fibers will start appearing. Then it will be continued with the metaphase 1. You can see that in the metaphase 1, each bivalent will be attached to the opposite pole. Each bivalent is having 2 chromosomes. One chromosome is attached to the one pole only. Another chromosome of a bivalent is attached to the opposite pole. So this is an important point. So one chromosome is attached to only one pole, not to both the poles. That is the difference between mitosis and meiosis. So in metaphase 1, all the kinetochores will be placed on the surface of the centromeres and the spindle fibers will come and attach to the kinetochores of the bivalents. And they'll bring the bivalent chromosomes to be aligned on the equator to form the metaphase plate or equatorial plate. These are the events which will happen in metaphase 1. Let us see the brief events of prophase 1. This is the longest phase, prophase 1. So in that leptotin. In this the events will be beginning. The process of condensation will begin. Condensation of chromosomes will occur. Next one, chromomeres are seen in this. Zygote, process of pairing. called synapsis occurs here. Due to synaptinemal complex proteins. Results in the formation of bivalents. Remember, one bivalent is equal to 2 homologous chromosomes. So bivalent will be looking like this, with 2 chromosomes, 2 homologous chromosomes. Packaging, this is tetrad stage, where each bivalent will be showing 4 chromatids each. These 2 are called sister chromatids. If chromatids belong to 1 chromosome, they are called as sister chromatids. If the chromatids belong to 2 chromosomes, they are called as non-sister chromatids. And here, recombination nodules are formed. With the help of recombinase enzyme, crossing over occurs. They exchange the genetic material between these 2. Chromosomes will be exchanging their genetic materials. So, exchange of genetic material which occurs between the non-sister chromatids of a bivalent of homologous chromosomes pair is called as crossing over. This leads to variations in a species because this results in recombination, shuffling of genes. And finally, it leads to variations which are important source for evolution of a species. So, the major event in prophase 1 will be the packaging, where the crossing over occurs and it leads to reshuffling of genes, nothing but recombinations, and finally leads to the variations important for the evolution of a species. Next one, diploting. Here, due to the further condensation and dissolution of synaptinemal complex occurs. Dissolution of synaptinemal complex. And points of crossing over, X-shaped. Points of crossing over are seen here. And they are called as chiasmata. Chiasma singular, chiasmata plural. So, X-shaped points of crossing over, they are called as chiasmata. They are very clearly seen due to the dissolution of synaptinemal complex in diploting, and they are further undergoing the condensation process. Diakinesis. In this, terminalization of chiasmata occurs. Movement of chiasmata towards the ends of the chromosomes. And here, nucleolus, nuclear envelope, Golgi complex, and ER disappears. Spindle fibers are formed. So, by the end of this diakinesis, the chromosomes would have been released into the cytoplasm. Now, these are the basic events of liptoting, zygoting, packeting, and diploting and diakinesis. You can see here, condensation of chromosome begins. They start pairing up each other. Synapsis begins. Chromosomes are fully synapsed and crossing over occurs here in packeting. Synaptinemal complex is dissolved and the chiasma are visible. And further condensation of chromosomes will happen in diakinesis, and terminalization of chiasmata happens. You can see that color exchange happened in the packetine and it continued further. Now meiosis I, in that we have completed the prophase 1. Next metaphase 1, all the chromosomes are arranged on the equatorial plate. Then in anaphase 1, the homologous chromosomes which are placed on the equator, the bivalent, they get separated. Segregation of homologous chromosomes occurs in the anaphase 1. Segregation or separation of homologous chromosomes occurs in the anaphase 1. Because of a bivalent, one chromosome will move towards this pole, another chromosome of a pair will move towards the opposite pole. That happens in anaphase 1. In telophase 1, the chromosomes which have reached the ends already, they would have undergoing the decondensation process and they form the chromatin one more time. But this chromatin will not reach the fully extended state. They will not be too much elongated like the interphase. They will be somewhat like 25% extended than the telophase state. That's all. That will be there in this. And after that, nucleolus, nuclear envelope, they reform, Golgi reappears, ER reappears. And then after this, cytokinesis will be done, which divides the cytoplasm of this cell into 2 daughter cells. So by the end of this, if you have considered here the 4 chromosomes, if we are taking, 4 chromosomes are taken in this pic. So 4 are there, 1, 2, 3, 4. Here also 4 are there. But here by the end of the anaphase 1, at each pole you are getting only 2 chromosomes. So in telophase 1, you are getting in each nucleus 2 chromosomes. It is the same thing which is continued here. So when you take 2n is equal to 4 chromosomes when you start the meiosis, by the end of the meiosis 1, cytokinesis 1, each daughter cell is getting n is equal to 2 chromosomes only. That is why this meiosis 1 is called as reductional division. The actual reduction will happen. And meiosis 2, it is similar to the mitosis. So this is equational division. So prophase 2, metaphase 2, anaphase 2, telophase 2 are exactly similar to the mitosis only. You can see that here 2 chromosomes we have taken. It will not reach the fully extended state. But in between meiosis 1 and meiosis 2, we have one phase called as interkinesis. Interkinesis. In interkinesis, there is no DNA replication. Just it will be taking time for the preparation of the cell division. There is no DNA replication that happens in the interkinesis. So first meiosis 1, then interkinesis, then meiosis 2. So by the end of the meiosis 1, we are getting 2 daughter cells and they will be entering into the interkinesis. Later they will proceed to the meiosis 2. And meiosis 2 is again divided into karyokinesis 2 and cytokinesis 2. By the end of the meiosis 2, we are getting the 4 daughter cells which are haploid. One daughter cell will be forming the 2 daughter cells which are haploid. Second daughter cell will be forming the 2 more daughter cells which are also haploid. So this is the equational division. You can see here meiosis 2 is divided into karyokinesis 2, karyokinesis 2 and cytokinesis 2. Karyokinesis 2 is further subdivided into prophase 2, metaphase 2, anaphase 2 and telophase 2. So in the prophase 2, that chromosomes we have taken, there were in the previous picture, 3 were there. So we have started with the 3 chromosomes, complete chromosomes, because they are having the 2 chromatids each. You can see that 2 chromatids are there for each chromosome. So we are starting with a cell with 3 chromosomes in meiosis 2. In prophase 2, you know the events like nucleolus, nuclear envelope, Golgi ER disappears, centrioles will move towards opposite poles and chromosomes are released into the cytoplasm. Then the spindle fibers will come in the metaphase 2. They will come and attach to the kinetochores. In this metaphase 2, each chromosome is attached to the 2 poles by 2 different spindle fibers. You can see this. This is the similarity of the mitosis and the meiosis 2. So here each chromosome is attached to both the poles. Whereas in meiosis 1, each chromosome is attached to only one pole by 2 spindle fibers. But here each chromosome is attached to 2 spindle fibers coming from opposite poles to the 2 kinetochores. And these chromosomes will be brought to the center of the cell and they are aligned in the center to form the metaphase plate. In each daughter cell, this happens in the metaphase 2. In anaphase 2, here centromere splits. Exactly the centromere will be splitting in the center and it results in the formation of daughter chromosomes. And the daughter chromosomes are now having only one chromatid each, and these will migrate towards the opposite poles. Because of the contraction of this side spindle fiber, this daughter chromosome will be migrating towards this side, and this daughter chromosome, which is formed from this chromosome, will be migrating towards the opposite pole. So the segregation of the factors, okay, will happen in the NR phase II, and finally the decision which gamete is receiving which factor, that will be decided by the NR phase II. But actual segregation of Mendelian factors or the genes, that happens in the NR phase I. So by this centromere splitting, daughter chromosomes are formed and they migrate towards the opposite poles. In telophase II, the 3 daughter chromosomes which are migrated towards one pole, they will be covered by the nuclear envelope and nucleolus, and then it forms the 2 daughter nuclei in this. After that, cytokinesis 2 will happen and it divides the cytoplasm either by cleavage furrow or by cell plate method to form 2 daughter cells from each daughter cell. So ultimately, as we have 2 daughter cells at the end of the meiosis I, meiosis II will be performed for 2 daughter cells, and by the end of the meiosis II, we will be getting 4 daughter cells each. You can see that this is one cell, this is second cell, and ultimately we are getting here 2 cells and here 2 more cells. So by the end of the meiosis II, we are getting the 4 daughter cells, and all these 4 daughter cells are haploid in nature. And here we have started the prophase II with n is equal to 3 chromosomes, and each chromosome is having a structure like this. And finally, in the meiosis II, each chromosome will be having this kind of structure, and n is equal to 3 only. So it remains same, n number 3, it remains same there also, but here the chromosome is having 2 chromatids. Here each chromosome is having only 1 chromatid. That is the difference. Significance of meiosis: it maintains the constant chromosome number. Maintains constant chromosome number of a species throughout generations. From one generation to the next generations, it will be maintaining that. See, we have human beings, even though several generations when you are taking also, 2n46 will remain as it is. Because the gametes are formed by the meiosis and they are getting fertilized and they form the zygote. So 2n number remains constant throughout the generations because of the meiosis. Next, it leads to variations. You know, these variations are the result of crossing over recombination process, which are important for evolution of a species. Definitely the siblings are different, the offsprings are different from the parents because of the meiosis, because it leads to the variations. So these two are the significance of meiosis. Next, the questions. The first one, if there are 60 chromosomes in S phase, then what will be the number of bivalents in zygotine stage? 60 chromosomes are there in S phase. In S phase means obviously the number of chromosomes will remain constant, only DNA will be replicated. So 60 chromosomes in S phase, they will be looking like this with 2 chromatids each. And what will be the number of bivalents in zygotine? Always the zygotine bivalence will be, there is a formula, this is equal to n. So if 2n is equal to 60, what will be n? It is 30, because 30 pairs will be there. Zygotine always is represented by the bivalence, a pair of homologous chromosomes. So n number will be nothing but bivalence. Bivalent number is equal to n number of that cell. So here they asked bivalence in the zygotine. So 60 chromosomes are there like that, and bivalence will be a pair. So for this question, the correct answer will be 30. Number of bivalents will be just 30. Next. To produce one or two pollen grains, how many meiotic divisions are required? There is a formula for this, for the meiotic divisions, meiosis, n by 4. So here the number of pollen grains are 102 by 4. It will become 25.5. But we can't decide that the division is incomplete. Half of the division is completed, half is not completed. We can't take it. So we have to round it off to the next nearest number. So instead of 25.5, the 26 will be correct, because the division cannot be stopped in the middle unless and until you are using any mitotic poison or meiotic poisons like that. So we can't take that. So how many meiotic divisions are required to form one or two pollen grains will be 26. Though the number you are getting 25.5, 5.5, 5 you have to round it off to the next nearest number, it will be 26. All are essential stages that takes place during meiosis except. All are happening in the meiosis except which one? You know that terminalization of chiasmata, it happens in diakinesis. Nuclear division and cytoplasmic division occurs twice. Yeah, because karyokinesis 1 and karyokinesis 2. Cytokinesis 1 and cytokinesis 2. But DNA replication occurs only once. Number of chromosomes after meiosis II in each cell is reduced to half. After meiosis II, it is reduced to half and DNA amount remains the same. Okay. So, the number of chromosomes after meiosis II in each cell is reduced to half. This is correct only. And DNA amount remains same is wrong, because DNA is also reduced to half. Let us take for example, if a cell is having 2n is equal to 46 chromosomes. I'll take the example of human cell only. 2n is equal to 46. So, in this 2n is equal to 46, after the end of the meiosis 1, each daughter cell will be getting n is equal to 23. After meiosis 2, each daughter cell will be getting 23. But coming to the amount of DNA, here if the amount of DNA is 4C in G2 phase, then after the meiosis 1, each daughter cell will be having the DNA amount of 2C. Here each daughter cell will be getting just 1C because 4 daughter cells are formed here. So DNA amount is also reduced to half, and it remains if you take the G1 stage and S stage. In G1, 2C will be there. In S, it will become 4C. So now the DNA content, if you consider with the G1 also, it will be 1C. It is reduced to half. So this option is wrong. So number of chromosomes after meiosis II in each cell is reduced to half. Correct. And DNA amount is also reduced to half. This is correct for this. Next one. Prophase 1 is subdivided into 5 phases based on chromosomal alignment, chromosomal movement, chromosomal structure, chromosomal behavior. It is subdivided into 5 phases based on the behavior. So, for this, correct answer will be chromosomal behavior. Next one. Diplotin can last for months or years in oocytes of all vertebrates. This is wrong. Oocytes of all invertebrates. Wrong. Oocytes of some vertebrates. Correct. Oocytes of some invertebrates. It is wrong. So, for this, the correct one is the oocytes of some vertebrates. Segregation of Mendelian factors, this is small a, capital small a, occurs during diplotin, zygotin, anaphase 1 and anaphase 2, where they are getting actually segregated. Segregation occurs in anaphase 1 because they are getting separated like this. Homologous chromosomes are there. I am taking on one chromosome capital A and another chromosome small a, so they are getting separated in anaphase 1. So, segregation of Mendelian factors occurs in anaphase 1. How many chromatids are present at each pole in telophase 2 in a myocyte of onion? Myocyte means a diploid cell of onion. It will be having 16 chromosomes basically. And in telophase 2, they are asking at the end of the meiosis 2, telophase 2, there how many will be there at each pole? How many chromatids are present? You know that it will be having, by the end of the meiosis I, it will be becoming n is equal to 8. By the end of the meiosis II, also n is equal to 8 will be there. But they are asking the chromatids. So in that 8 also, 8 chromatids only will be there. Because the chromatids after the end of the meiosis I, they will be like this, and the chromatids at the end of the telophase II will be like this. So for this, the correct option will be 8. At each pole, because one side, one cell will be having one pole here and one more pole, other chromosome will be there. The total DNA content of each daughter cell is reduced during meiosis because chromosomes arms are lost during crossing over. Chromosomes do not replicate during the interphase preceding meiosis I. Chromosomes do not replicate between meiosis I and meiosis II. Half of the chromosomes from each gamete are lost during fertilization. Chromosomes do not replicate during meiosis I and meiosis II because in interkinesis, chromosomes do not replicate, or DNA do not replicate between meiosis I and meiosis II. This point is interkinesis. So the total DNA content of each daughter cell is reduced during meiosis because of this. As it is not replicating, it is reduced. That is the reason. Stage between meiosis I and meiosis II is called as cytokinesis, karyokinesis, interphase, interkinesis. Interkinesis. It is called as interkinesis because it is happening between two divisions. Number of chromosomal groups at equatorial plate of metaphase I of a plant body having 2n is equal to 50 chromosomes shall be. How many groups are present at the equatorial plate of metaphase I of a plant body having 2n is equal to 50 chromosomes? How many groups are present? Like groups means the bivalence only you can think. 2n is equal to 50. So how many groups will be placed on the equator means it will be 25 groups because bivalents they are asking. They will be placed on the equator. So bivalence will be n number. So it will be 2n is equal to 50 means n will be 25. First division is reductional and second division is equational. It is true for meiosis. Meiosis I, this is meiosis I and this is meiosis II. Correct? First division is equational and second division is reductional. This is wrong for meiosis. The best material to study meiosis is the root tip. No, this root tip is meant for mitosis study, not for meiosis. Anthers are used to study the meiosis. Meiosis maintains a constant chromosome number throughout generations. This is correct. So the first one and the last one is correct. Find the correct statements from above. It is A and D. So this is correct. This option is correct for this. In a cell, 23 bivalents are present in zygotine. That means N will be 23. What will be the number of chromosomes in daughter cells after meiosis I and meiosis II? So 2n is equal to 46. And you know that meiosis I, it forms n is equal to 23. And by the end of meiosis II also n is equal to 23 only. So they are asking after the meiosis I and meiosis II, how many number of chromosomes will be there? So for this, the correct option will be 23 and 23. But here the meiosis I, the chromosome will be having the structure with two chromatids after meiosis I. This is the structure of the chromosome after meiosis II. This is after meiosis I, this is after meiosis II. So the number remains same. Here also one chromosome in both the places, but here it has two chromatids. Here it has only one chromatid. That is the difference. So for this, the correct option will be C, 23 and 23. Genetic constitution of gametes is finally decided in. Where the genetic constitution will be finally decided? Anaphase II, metaphase II, anaphase I and metaphase I. The final decision of factors which are received by the gametes will be happening in anaphase I because segregation of Mendelian factors occurs in the anaphase I. Match the following, column 1 with column 2. Packetine, Zygote, diakinesis, diploide, nucleolus disappear, chromosomes pair up, tetrads appear, chromosomes move away from each other. You know that pachytene is meant for tetrads appearance and zygote is meant for chromosomes pair up. In diakinesis, nucleolus disappears and in diploide, the chromosomes move away from each other. So for this, the correct option would be B, 1C, 2B and 3A and 4 is D. Next, what will be the amount of DNA in each cell produced after meiosis II if a meiocyte contains 30 picograms of DNA in G1 phase? So in G1 phase, 30 picograms is there, the DNA content. So in S phase, it will become double, so 60 picograms. And it will be continued up to prophase 1, metaphase 1 until anaphase 1 up to here. And in each daughter cell, it will be getting only 30 picograms each. Again, if a meiocyte contains, so each cell after meiosis II. This is after meiosis I. But after meiosis II, it will become again further reduced to half and each daughter cell will be getting only 15 picograms because each daughter cell will be having a chromosome number reduced to half with only one chromatid. So it will become 15 picograms in each daughter cell after meiosis II. So whatever the DNA content you are taking in G1, in S phase it will become double. And S phase, it will be continued up to the anaphase 1. It is the same amount will be continued. And in telophase 1, the 30 picograms will reach one pole and 30 picograms will reach another pole. So one nucleus will be having only 30 picograms. So one daughter cell will be receiving after meiosis I 30 picograms. And after meiosis II, that splits up because of the centromere splitting in meiosis II, anaphase 2. The 15 picograms will be reaching the daughter cells. With this, we have completed the meiosis. Thank you.